This video will serve as a brief introduction to protein structure and function. Proteins are, of course, profoundly important molecules that play lots of different roles within living organisms. And we have all heard of proteins before. We know something about them. But this chapter will really focus on the basic structure of proteins. So let's begin with recognizing that proteins are nothing more than long chains of amino acids linked together. So this image you see here just illustrates some amino acids. So some of these may sound familiar. So phenylalanine, tryptophan, glycine, serine, aspartic acid. So these are all examples of amino acids. And so again, proteins are nothing more than a bunch of amino acids linked together. When we get amino acids linked together, we have some fairly distinct reactions taking place. A very general term or concept you might hear referred to is that of a condensation reaction. So condensation reactions illustrated here. So here we have a couple of amino acids located, one located here, one located here as well. And when these amino acids are linked together, essentially what happens is the carbon on one amino acid of part of this carboxyl group links up with the nitrogen on this adjacent amino acid that's part of the amino group. And when these two link together, what gets released? Well, two hydrogens and oxygen, they join together to form a water molecule. So that's kind of released as a byproduct here. And again, a bond formed between the carbon and nitrogen. We say that's a peptide bond, but really that's just an example of a covalent bond. So this carbon covalently bonds to this nitrogen, forming a peptide bond, essentially linking these two amino acids together. So proteins are nothing more than hundreds or thousands of amino acids linked together in a very specific sequence. So where then are proteins formed? Well, proteins tend to be formed within cells as a result of reactions that occur in ribosomes. So ribosomes are these small structures found within most living cells, and again, whose primary role is to produce proteins. You perhaps have heard of the general processes referred to as transcription and translation. So those are part of what we might call the central dogma of molecular biology. So in a sense, what this describes is our basic understanding of information flow within cells. So here's a typical eukaryotic cell. Here's a nucleus. Here's DNA as part of chromosomes within this cell. So when a gene is expressed, the sequence of bases in the DNA are read and used to produce a corresponding piece of RNA, namely messenger RNA. The messenger RNA then leaves the nucleus, interacts with a ribosome, as we see here, and the sequence of bases in the mRNA are read and used to produce a protein, shown in green here, which again is just a sequence of amino acids, and the sequence of amino acids in that protein, of course, ultimately is dictated by the sequence of bases in the gene that's expressed to begin with. So again, that's just this flow of information. Transcription, when DNA is used to produce messenger RNA, and translation, when the RNA is read and used to make a specific protein. So in essence, that's what DNA does. Its primary role is to code for proteins within organisms. So if we look at proteins, they're really quite complex in terms of their structure, and they, as a general rule, they have to have a very specific overall three-dimensional shape in order to function properly. So when we look at the structure of proteins, we can identify some basic levels of organization. So what are the levels of organization found within proteins? Well, let's first of all identify what we refer to as the primary structure of a protein. When we talk about the primary structure, all we are referring to is simply the sequence of amino acids in that particular protein. And of course, all proteins have different sequences of amino acids. That's what differentiates one protein from the next. So you might ask the question, well, what is it that determines the primary structure of a protein? Well, again, it's simply the sequence of bases in the gene, right, in the DNA that's expressed, right? So DNA determines the primary structure of proteins. We can also identify secondary structures within proteins. As examples of secondary structures, we often see these regularly occurring organizational units, namely an alpha helix and a pleated sheet. So the alpha helix sort of has this helical shape to it, as you can see here. Pleated sheet, much like corrugated cardboard, as you see here. Again, these are fairly common in many proteins, and they are relevant in many ways. For example, many proteins are found embedded within cell membranes, and often it's the helical region of a protein that actually is embedded within the phospholipid bilayer of membranes. So beyond the primary and secondary structure, we can talk about the tertiary structure of proteins. Tertiary structure of proteins just refers to the overall three-dimensional configuration of a protein. It's hard, of course, to illustrate on a two-dimensional format, but I just want to point out, again, the overall three-dimensional shape of a protein. That's its tertiary structure, and there are many types of bonds involved in the configuration of a protein. So you might have some ionic bonds holding parts of a protein together. You might have some covalent bonds, much like this disulfide bond right here. You can have all sorts of other interactions as well. But again, these are all important, right, in maintaining the overall three-dimensional conformation of a protein. So many proteins actually go one step farther, and instead of being configured of just one long chain of amino acids like we see illustrated in this green ribbon in this image, they actually consist of multiple chains of amino acids. So those proteins that contain many chains of amino acids, two or more, we say they display quaternary structures. So hemoglobin, a classic example. Collagen, it's another example as well. But notice what we're looking at here is a single molecule of hemoglobin, which consists of 1, 2, 3, 4 chains of amino acids. In order for hemoglobin to function properly, it has to have all 4 chains of amino acids present. Hemoglobin is, of course, an incredibly important molecule found within our red blood cells. It has the ability to absorb oxygen, right, typically when that blood is circulating in our lungs, but also release the oxygen in other parts of our body. And again, in order to do that, it has to have these 4 chains of amino acids present. And again, we just use this as an example of a protein that displays the quaternary level of organization. That concludes our brief introduction to proteins.